Bismillahirrahmanirrahim. Assalamu alaikum. Today the topic of discussion is functions and types of antibodies. The learning objectives for today's lecture are to study the structure and function of antibodies and then to study the role of antibody in different situations, to compare T-independent and T-dependent antigens, and finally to understand the different types of antibodies produced. Before discussing antibodies, let me give you a brief review of how these antibodies are produced. B cells are originated and they get mature in the bone marrow. Then these B cells are released into the blood and they keep on circulating between the blood, lymph, and lymphoid tissue. When these B cells recognize specific antigen, only then they get activated. This recognition of antigen occurs by a specific set of receptors which are present on the surface of the B cells. They are known as the B cell receptor or BCR. Once activated, these B cells will proliferate and they will form the plasma cells, which in turn will produce and secrete the antibodies specific to that antigen which was initially recognized by the B cell. Now let's discuss the antibodies. Antibodies are actually the glycoprotein molecules which are produced by the plasma cells in response to an antigen or immunogen. Their function is to protect the host against extracellular pathogen. So what is an antibody? Antibody forms the essential part of the adaptive immunity and they are also known as the immunoglobulin. They specifically bind a unique antigenic epitope that is also called as the antigenic determinant. Here it is shown in the red color. These antibodies possess the specific antigen binding sites and they constitute about 20% of the proteins in plasma. They can be monoclonal as well as polyclonal. This figure shows the basic structure of an antibody. The basic structure of antibody resembles a Y-shaped molecule having two antigen binding sites and a stalk known as Fc region. These antibodies circulate in the blood and lymph from where they reach to the site of the pathogen invasion. Then these antibodies bind to the pathogens or antigens and thus they will activate the defense mechanism which will lead to destruction of that pathogen. Antibodies are also known as the immunoglobulin and this is because they belong to a group of proteins known as the a group of glycoproteins which are known as the globulins. And the term immune reflects that these glycoproteins play a major role in immunity. The basic structure of all the antibodies and B cell receptors are same. The B cell receptors are also immunoglobulin. The difference is that they are the membrane bound immunoglobulins, whereas the antibodies are secreted immunoglobulins. So the antibodies can exist in two forms, either in secreted forms, which are known as the antibodies, and when they are bound to the cell surface, they are known as the B cell receptor or immunoglobulins. So what does the antibody look like? Let's now study the basic structure of the antibody in detail. It consists of four polypeptide chains, two identical heavy and the two identical light chains. The heavy chains are designated as H and the lighter chains are designated as L chains. As you can see here, the heavy chains are the larger ones and the lighter chains are the shorter ones. The term heavy and the light refers to their molecular weight. So this means that the heavy chains have more molecular weight as compared to these lighter chains. Now since these are the polypeptides, the amino acid terminal, this terminal, N terminal of this polypeptide chain is present at the tip and the C terminal of these polypeptide chain is present at the base of each polypeptide chain. As you can see in this image, these chains are assembled into a Y-shaped structure. Each light chain is attached to the heavy chain by means of a single disulfide bond, and the heavy chains are attached or connected to each other via two disulfide bonds in the mid region. Mid region of the antibody has a considerable flexibility, and this region is known as the hinge region. The main advantage which this region provides to an antibody molecule is the ability to adjust to different arrangements of antigens and antigenic epitopes. In other words, this hinge region makes possible the rotation and bending of this antibody molecule. So this is the variable portion of this antibody molecule, and this is the constant portion. The stalk of this Y-shaped molecule is the stem region, which is known as the Fc portion. To each heavy chains of the carbohydrate molecules are also attached. And these carbohydrates may serve many additional functions, such as they may increase the solubility of the immunoglobulins. Now let's briefly discuss the light and heavy chains. L chains, as also said before, these are the two shorter subunit of this basic antibody molecule. And each light chain contains about 220 amino acids. In humans, there are two types of the light chains, lambda and kappa. Both these lambda and kappa chains are similar in structure and function, but they are encoded by different genes. But it is very important to note that each antibody molecule uh, produced by a B cell will either have kappa or a lambda chain, but never both. The clinical implication of these light chains lies in that fact that the distortion in this ratio between the kappa and lambda chain may be used to diagnose the immune system abnormalities, for example, lambda producing B cell tumors. Now, the heavy chains. Eight chains are the subunits of this antibody molecule. Each H chain contains about 440 amino acids. There are about 5 types or subclasses of the heavy chain in humans, and these are designated as IgG, IgM, IgA, IgD, and IgE. And their distinctive functional properties depend on their Fc portions of the heavy chains, where it is not associated with these smaller or the shorter L chains. Each heavy and light chain contain two distinct regions, variable and a constant region. So this is the variable region. The variable region of the antibody, this is the actually located at tip of this antibody, and the function of it is to bind the antigen. Specificity of the antigen binding is determined by this portion, variable portion of both the heavy and lighter chains. Now the constant region. The constant region forms base of this antibody molecule, and this constant region can bind cell receptors and the complement proteins. Now the epitope or the antigenic determinant. These are actually that portion of an antigen which combines with products of the specific immune response. Like here you can see that the red ones here indicated, these are actually the epitopes or the antigenic determinants which will then bind to products of immune system or antibodies. Now the antigen binding sites, this shows the diversity of N-terminal that can bind to a number or a variety of the antigenic determinants. You can see here the shape of these epitopes. They all are different, and the antibody variable sites, they will be specifically, specially they will be designed for these epitopes. Now they're coming towards the function of antibodies. Antibodies help in neutralizing the toxins and viruses. They opsonize the microbes. They prevent attachment of microbes to the mucosal surfaces. They can activate the complement system, and they provide the antibody-dependent cell-mediated cytotoxicity. So this is a summary of the all the subclasses of these immunoglobulin or antibody. The structure of IgG is a monomer. In case of IgM, its structure is a pentamer. 5 molecules are attached by means of this J chain. And this immunoglobulin is dimer with a secretory component in case of the IgE, which are joined by the J chain. In case of IgD and IgE, these immunoglobulins are the monomers. The highest percentage of these immunoglobulins are the 80%. These are the IgG, which are present in the serum. And the next one are of the IgA, 10 to 15%. IgG are present in the blood, lymph, and intestine. IgM in blood, lymph, as well as on the surface of B cell. IgA are present in the secretions such as tears, saliva, mucus, intestine, milk, blood, and lymph. And IgD, they are present on the surface of B cells, blood, and lymph. And the Ig are present on within the mast cells and basophil cells. The molecular weight of IgM, it is the highest among others. And the maximum half-life in serum is 23 days, that is of the IgG. IgM and IgG leads to complement fixation, and the IgG can be transferred via placenta. IgG enhances the phagocytosis. They neutralize these toxins and virus, and they protect fetus and newborns. IgM are especially effective against the microorganism and agglutination or agglutinating the antigen, first antibodies produced in response to the initial infection. IgA, they provide localized protection on mucosal surface, and the IgG function in the serum are still not known. And their presence on the B cell function in initiation of the immune response. The IgE class are involved in the allergic reaction, and they possibly can lead to lysis of the parasitic worms. Students, this table is very important from your examination point of view. You must memorize this. So these are the subclasses of the antibodies: IgG, IgA, IgM, and IgD and E. And these are their main function. Kindly go through this table. Now coming towards the antigens, T independent. These activate B cells without the help of T cells. These are actually the polysaccharides. Their structure is characterized by multiple copies of the same antigenic determinants. And the examples include pneumococcal polysaccharides and the lipopolysaccharides. They can induce IgM immune response, but they do not cause the Ig class switch. Ig class switching is actually a biological mechanism that changes or switching of one isotype of antibodies such as the IgM to IgG. The antibody will retain affinity for some antigens but can interact with the different and effector molecules. These will induce the immune response on second exposure with the same intensity as that was in the previous exposure. Now the antigens which are T dependent. These antigens require they require the help of the T cells to activate B cells. These are actually the proteins, and structurally these antigens are characterized by a few copies of many different antigenic determinants. Examples include the microbial proteins. So this is the summary of functions of these antibodies. When the bacterial toxins are introduced, so what does these antibodies do? They will bind these toxins and they will neutralize it, and they will prevent them from binding to the cell receptors. And thus these bounded antibodies with toxins, they are ingested by the macrophages. When the bacteria are present in the extracellular phase, the macrophages will opsonize. In addition to, initially they will be contacted or connected with these antibodies, and then they will be opsonized, and finally they will be ingested by the macrophages. When the bacteria they are present in the plasma, they will cause the complement activation, and as a complement activation, these antibodies will bind to these pathogens, and as a result of this complement activation, finally there will be these will be lysed and ingested. That's all for today. Thank you very much.